In KEFIS, we have several labs. We have one at headquarters, another one at Plant Health Quarantine that is in Muguga, another one in Nakuru, and also another one in JKA in Mombasa. So this is our complex laboratory that we have in KEFIS headquarters, and you can agree with me, it is complex. And also I can tell you for sure it has all the equipments that are needed for us to run the testing. Now, one of the things that we do here, it houses molecular lab, and in the molecular lab, that is where we have the GMO testing, we have the real-time machine, we have the conventional, we have the ELISA, and also we have the rapid kit. You're going to see it in my presentation. We also have for diseases, for the plant health, for food microbiology, and analytical chemistry. Now, there's one person who asked whether we do surveillance. Yes, we do surveillance even as KEFIS and NBA. Sometimes we do in collaboration with NBA, and sometimes we do individually. And why do we do this surveillance? We do them so that we ensure that our permits are not misused by those people who we have given permits, so they are not used to bring unauthorized GMO. We also do surveillance to ensure that no unapproved genetic element are released to the farmers. Remember, we are the National Plant Protection Organization, where we protect our farmers from anything, from anything that is not right. Then we have the variety release. The variety release is where we test the genetic purity and conformance of the biosafety guidelines. Now, for example, we have the BT cotton that has been approved. We usually look at the genetic purity because what they tell us is, or what the scientists have confirmed is that BT has, is against the caterpillar. So for us, we have to totally confirm genetically and also the seed department morphologically when they are doing the post-release that indeed it can be able to kill the caterpillar as they have told us. And then we have the border surveillance. The border surveillance, we do it with the National Biosafety Authority because where they are not there, we are there, and when we are not there, they are there. We keep each other guarded. So for the surveillance, as I've said before, and as my colleague and also boss has said, is that before you come to KEFIS for a phytosanitary certificate, you have first to go to National Biosafety Authority in order for them to give you a clearance. So that is how we work in harmony. If you're bringing anything that is genetically modified, first to go to NBA, they clear you, then we accept. If you're not cleared by NBA, we will not accept you. We will not accept to proceed with your either research or even bringing the seeds into the country. We also make sure that we regulate and monitor all the research that is going on together with National Biosafety Authority to make sure that they have complied to the laid down procedures. Now, let me come to the detection that we usually do in the lab. You are able to get the the gene in two categories. One, it is in protein, and the other one, it is in the DNA. And the methods that we use, or the detection methods that we use, we have the detection or qualitative analysis. That is a yes or no, a positive or a negative. So when we run the sample at random, or sometimes we run them in the laboratory, we are able to know whether it is positive or not positive. Then we have the identification part of it. The identification part of it is the one that tells us which gene is that. Remember, we have authorized, or we have, the BT cotton has been commercialized. But what has it been commercialized for? It has been commercialized against the caterpillar that they call the bollworm. So nothing else has been commercialized for the BT cotton. So we usually check, are you bringing cotton that has been commercialized, but by what you told us, or what we gave you the authority, what we gave you the permission to bring in, or something else. And then we have the quantitative analysis. The quantitative analysis is where we get the percentage of the GM that has been modified in that particular product. What we do in the laboratory is we use the PCR test, where we have the promoter gene, we have the transgene, and also we use the terminator and the selector gene. Now for the promoter, it has to be there. For the gene, it has to be there. For the terminator, it cannot be there, because as the science goes, the terminator was already eliminated. So if we find that there's a terminator in that product, then it raises alarm, and we have to follow it up together with our colleague from the National Biosafety Authority. Of course, we have kits that we have that can be able to determine these elements. Now, this is one of the methods that we use, and very soon it's also going to be used. Actually, not very soon. Right now, it is being used by our border people, that is the lateral flow strip. The lateral flow strips work very well, just like the HIV testing. You know, when you take the HIV testing, the strip that is there, you put a drop of your blood, an antigen is put on, and then it flows for about 5 minutes, and then you are able to get whether you can get 1 strip, 2 strips, or no strip. So that is the same way lateral flow strips work. If you get 1 strip, that is okay, or 1 line, that shows that it is a control. If you don't get 1, if you get 2, that is a positive one. And the people at the border, they are able to do that, and they can bring that sample now to the lab for confirmatory. If you get no strip, then that is invalid, and a result has to be redone. This is what I'm saying. There's a test strip, there's a red control line, there's the test, there's the sample, and then you have the positive that shows the 2 of them, and then you have the negative. But if you get one that does not have anything, then that shows that kit has either expired or it is invalid for that process. We also have another one that uses protein. This is ELISA. And I know maybe for those ones who are scientists know that it is used as protein. As you can see, okay, I can't get the pointer. Okay, as you can see, there's a place where it is plain. That shows that it is negative. When you run your test and you can see the purplish part of it, it shows that it is positive. Even the yellowish part of it, it shows that it is positive. So you have to go for the confirmatory part of it, which is PCR. This kind of protein-based analysis, they are very simple. They can be done by anybody, even you, by the way. If you have the strips for the GMO, you can be able to use them. The only thing that I think you have to look for is, what is it called, distilled water or grade water that you have to put, because you cannot put the tap water, it will contaminate. But it is very simple to run. It is very cheap. Okay, cheap is relative, but it is cheap. It is reliable. It can give you accurate results. It gives you within very few minutes. So if there's a product that you're finding in the market and you think that is GM, it's something that you can do for that one. Then we have the DNA-based detection. For the DNA-based, it's a bit involving, because what we do, we go through the process of DNA extraction, and then after doing the DNA extraction, we quantify that DNA using an instrument called nanodrop to make sure that you have the DNA, and then after that you go to the PCR to run the process to find whether it is positive or negative. So we have 2 different types of PCR. We have the conventional PCR, and you have the real-time PCR. So for the conventional PCR, it will give you something that is called the bands. As you can see them, there's a GMO negative part of it, and there's a GMO positive part of it. So when you see those bands, we always know that it is positive, and we go for more confirmatory. GMO analysis is a process-based. Once you get the GM in the field using the lateral strips, you come to the lab with it if it's positive, then we do the extraction of the DNA, then we come to the conventional part of it. We find that one, it has shown that it is positive, we'll go to the PCR, the real-time one that I'm going to show you in a minute. So this method shows not only qualitative analysis, that is a yes or no, but we are able even to get the quantity part of it that has been modified. So this is our PCR real time, and it is in our laboratory. We have 2 types, the conventional one, and also we have the real time. All of them will be able to give you the analysis for the results for the GMO. Please note that for the real time one, it can be able to detect up to 0.01% of any modification. So even if it's the one that the NBA, remember Moshiru was talking about the contamination, we'll be able to differentiate whether it is contamination or it is scientifically done. So once we do the DNA part of it, the real-time PCR, it shows the curves. Yeah, I'm the one who was taking it back. It shows the curves, and these curves, I can now show you well where the way they show. It is like a sigmoid curve that it analyzes. And when you look at the graph from 0 up to around 10, 11, 12, 13, there we don't expect any results, any amplification or any, the curve to start showing. But from 14 up to around 34, that one we now start knowing that it is indeed modification. If it comes to the very far end from 36, 38, and 40, we can assure you that it's going to be less than 1%. And we all know the biosafety rule that says that if it's below 1%, then you don't consider it as GMO. So those are the curves and the percentage. This one is usually done using the standard deviation method together with the PCR, the cycle numbers. So we usually calculate it in the laboratory. So apart from doing all that in the lab, we usually have controls that we have. When you're running any analysis, we make sure that we have a positive control, we have a negative control, we have an environmental control, we have a blank control, so that you are accurate, that your results are completely the way they are supposed to show. So if it's supposed to show positive, positive. It's supposed to show negative, negative, but there should be no invalid. So those are the positive, negative, and no template control, to make sure even your environment and the working area that you're working with, it is purely conducive for you to run the test. Thank you.